2. We're going to do enero. 1. This would be E. coli.
Kid sample 1.
Right.
For this one, I fully intend to make a plan about like joke about like responsibly sourced strep or something like all organic.
How cool the patterns are on that one.
I know,
That's
right?
from Talon's Up the Creek one.
Yeah,
Do you recommend with flash or no flash?
probably without it. There's enough overhead light. I think we're okay, but you can experiment a little bit.
No flash.
So the students learned all this technique about how to apply the sample of bacteria to the agar. And agar is made of seaweed.
Nope, wrong. Oh, that wasn't to you, that was to me.
Oh.
You are correct.
I think we might have to harvest some seaweed to make agar in this place. So this entero one is the one you thought was contaminated, right?
Yeah, I'm gonna guess that large circular kind of umbenet looking one, which means it has a big bump in the middle.
Umbenet.
Umbenet is probably some staph, whereas the cool looking ones down at the end are called rhizoid, when they have those little fingers coming out of them.
Okay, I'll come back to the Visinate ones.
Yeah.
Those are the...
So these little guys are rhizoid with the little fingers that are sticking out there. But there's probably, if you were to look at it underneath the microscope, you'd probably see some filamentous ones as well. It's harder to tell.
Okay, then we got an E. coli too. I'm not sure who did which one, but there's some of the. Oh, there's some filamentous. Is that filamentous or is that just shiny?
Just shiny, but we'll be able to tell
Okay.
really. I think there's 2 different colonies there, because there's one that's the rounded one here, and then one that has more of a central growth where it's fingering out. I'm guessing this is more filamentous if you were to look under it under the microscope. And then the throat swabs have a nice combination. You have some clear white colonies, which are probably the strep, plus some yellow ones there that can be maybe staph or something else. But the really fascinating thing about this plate is how well it cleared out how much hemolysis you see. And you can see all the way through, so it's beta hemolysis. You can see all the way that it's taken all of the color out and left it yellow rather than green. Whereas this one, there's thousands and thousands and thousands of colonies on there, but you don't get that same hemolysis. What you see is more of a greenish color to it, which is more alpha hemolysis, where you just get a little bit of the blood shoot up, but not enough to kind of clear it out. And you compare that color to one of our plates that has almost nothing, and you can see you still have this, yellow-red color here, where this has more of a greenish hue, and this one has a completely clear. So the blood agar will change a little after it's been in the incubator for as long as these guys were. Cool stuff. So just looking at this was one of our enero plates, and we think this is probably a contaminant because it has this nice blown out hemolysis. But if you look underneath there, you've got this collection of almost individual little pearls that have just piled up and piled up and piled up. And you get this lovely kind of what I think Connie described as fluffy-looking appearance to the colony.
Pretty sure she said bubbles too.
And she said it looked bubbly as well. But I don't know if you could take a picture through the lenses. Sometimes you can, but if you get it right over there, it might adjust to it, but I doubt it. Yeah, it's probably not going to focus enough for you to do it.